The first mistake I've heard, one of the most common, if not the most common, is to equate or confuse epigenetics with the environment. This error probably comes from the introduction of the term epigenetics by Waddington. Some problems that epigenetics has faced, besides the difficulty of understanding the molecular mechanisms and the biological implications, include the lack of a clear definition, which has changed quite a bit over time. It was introduced by Waddington in 1942, referring to any environmental factor that was responsible for generating different phenotypes from the same phenotypes. She called them epiphenotypes, contrasting. Molecular epigenetics, the science studying molecular mechanisms regulating gene expression without genomic changes. Unlike Waddington's term, epigenetics is negatively defined, adding ambiguity. Thus, it is less precise than positive terms, as it covers gene expression regulation without DNA sequence changes. In contrast to the first term introduced by Waddington, the definition of epigenetics is a negative definition. It will never be as precisely defined as a positively framed term, since epigenetics includes all regulation of gene expression without changes in the DNA sequence. It lacks precise definition, like a positive term, as it regulates gene expression without DNA changes. This ambiguity has allowed errors to persist. Epigenetics and environment are distinct. And it's possibly that ambiguity, which has historically persisted in the definition itself, that has allowed some conceptual errors to remain present even today. So epigenetics is not the environment, nor is all the environment epigenetics, and not all epigenetics is the environment. There are intrinsic factors to epigenetics, or at least factors that are independent of the environment, such as age, gender, or genetic makeup. The environment isn't defined very precisely or in a very agreed upon way either. It generally refers to environmental toxins, types of pollutants, the duration and concentration, the source, and the route of exposure. But really, the environment includes other characteristics, some of which are personal choices that make up our lifestyle, including diet, exposure to alcohol or tobacco, and exercise. And some of us have had little or no choice, like the position of the uterus, whether we engage in more outdoor or indoor activities, or even the occupational risks we might face at our jobs. What really gets confused is the epigenome with the exposome, and the exposome is a term that was introduced relatively recently, in 2005, by Wild, and it refers specifically to all the environmental exposure that a human experiences from the moment they are born. So it's about a history of environmental exposure. He defines 3 overlapping domains: a general external domain, which consists of the natural environment, but also includes the built environment and even the psychosocial environment, economic environment, and the educational environment, as they all have effects on health. In fact, there are branches of epigenomics that study the psychosocial or educational contributions to the epigenome, a specific external environment which would be made up of our more or less free choices, our lifestyle, our diet, our physical activity, and whether we expose ourselves to tobacco or alcohol. Again, internal environment, which includes the epigenome and the genome, but also the rest of the omics. These 3 environments interact with each other and allow us to assess the predictive risk associated with the development of a disease throughout an individual's life. But environmental factors can influence several different levels, which is known as the central dogma in molecular biology. The central dogma in molecular biology, it's a concept that's expressed here in a reductionist way to make it easier to understand, illustrating how information is transcribed. DNA is transcribed into RNA, which is then translated into proteins that are responsible for metabolism, ultimately leading to the production of phenotype. But as I mentioned earlier, not all environmental factors affect at the same level, or necessarily at just one level. For example, some factors like oxidative stress, heavy metals, or prolonged stress fundamentally affect the genome and the epigenome, producing genotypes and epigenotypes, which is what genomics studies. Others, any effect that the genome has on the transcriptome also produces effects on it. Another, such as the ionizing reaction, fundamentally affects the proteome by generating proteotypes, which is what proteomics studies. And others, for example, endocrine disruptors or polycyclic aromatic hydrocarbons primarily affect the metabolome, producing metabolotypes, which is what metabolomics studies. The higher we accept this sequence of transmission of effects, of transmission of information, the more levels will be affected. But as I said, it's not necessarily the same level that gets affected, depending on the environmental factor. Oh, sun. So the environment is not epigenetics. What is epigenetics then? Well, I think at an introductory level, rather than diving into the molecular epigenetic mechanisms that are hard to explain and also hard to understand, it's better to see it visually. We already know that DNA is this double helix structure made up of two opposing nucleotide sequences that twist in a clockwise direction. They are found in a highly compacted state in the nucleus. It's called chromatin. We can distinguish three levels of compaction. The first level is the nucleosome, which you can see here, and it can be organized or compacted into a higher order structure, which is the 100 nanometer fiber. The nucleosome is made up of exactly 146 base pairs that wrap around an octameric core of histones, which are 8 proteins called histones, and they give that very characteristic beaded necklace appearance when chromatin is viewed under a microscope. Finally, the 100 nanometer fiber can be organized into the most condensed structure, which is the chromosome structure, and that's generally how we visualize genetic material. Although it's only visible during metaphase, well, epigenetics allows for the regulation of gene expression. One of the mechanisms it uses is to affect degree of condensation of chromatin by adding chemical compounds to specific residues in nucleotide sequences. These are some epigenetic marks, and the set of epigenetic marks is called the epigenome. An epigenetic mark can be of the activating type, meaning it leads to a more relaxed state of chromatin, less compacted, which allows access to the transcriptional machinery, and thus makes the gene transcriptionally active. It would be an epigenetic activation of the gene, or it can be an epigenetic mark of the silencing type that leads to a more compacted, less relaxed state of chromatin, which prevents access to the transcriptional machinery, and thus epigenetically silences the gene. So acetylation is an epigenetic mark always associated with activating types because it increases the repulsion force of nucleosomes, favoring those less compacted or less dense states. On the other hand, the addition of methyl groups, methylation in sequences, is considered a repressive type of epigenetic mark, meaning it favors less relaxed, more compacted states that epigenetically silence genes. In any case, the set of epigenetic marks determines the degree of activation or repression. Epigenetics is responsible for producing all the different cell types from a single genome in multi-tissue organisms. And the tissues also need to maintain their tissue identity through selective gene expression, mitotically. So any internal or external factor that affects that epigenome will condition or predispose or increase the susceptibility to diseases related to the generation of aberrant epigenetic patterns. That's epigenetics in general. And in a nutshell, too. The second conceptual error has to do with evolution and is about attributing some characteristics of Lamarckian selection to genetics. Lamarckian selection, which I think we've all heard of at some point, is the idea that tall trees induce the elongation of giraffes so they can reach food in the trees. Remember that Lamarckian selection is a selection directed by intentionality in evolution, so it's the environment that induces advantageous adaptive changes, and also promotes a transgenerational maintenance of those changes. The environment can cause changes in genetics, variants, or polymorphisms, and also in epigenetics, the variants. But it's not just the environment, the stochastic noise itself, the molecular noise from the genetic or epigenetic machinery responsible for maintaining genetics or epigenetics is also capable of producing or introducing those variants. In the case of genetics, these changes are rare, random, meaning they are almost irreversible. In the case of epigenetics, they can respond to signals, but they are not specific loci, they are not coordinated, they are dynamic, irreversible, meaning they are not directed as Lamarckian evolution proposes, nor is there any evolutionary interaction. They don't necessarily have to be adaptive, meaning they can be adaptive but also maladaptive. Some epivariants actually lead to a loss of resilience. Resilience is what is known as epigenetic toxicity, while others can lead to an acquisition of resilience, which is referred to as adaptation or epigenetic plasticity. This can result in beneficial short-term effects, but also more persistent effects over time. Also remember that for a phenomenon to be considered evolutionary, not only must changes occur, that have some adaptive advantage, but those changes must also undergo selection pressure and be maintained transgenerationally. And it's this transgenerational epigenetic inheritance, at least in mammals, that is quite a speculative topic right now. And if it occurs, it would be limited to specific moments and as a consequence of failures in the regulatory mechanisms that maintain epigenetics. Obviously, an epigenetic pattern that leads to an adaptive advantage has evolutionary implications. Even if it doesn't get transmitted transgenerationally because it might affect survival rates or reproduction rates, but it can't be considered evolutionary.